Welcome to this explainer. Today we're diving right into a seriously fascinating biological mystery, the precise minute-by-minute mechanism of how sweet potatoes actually heal their own wounds. The microscopic drama taking place just beneath the surface of this root crop, it holds absolutely incredible implications for global food security, farming tech, and the whole future of agricultural science. So, let's just jump right in. Okay, so to understand why this matters, you have to realize the scale of the problem. It is massive. Get this, in developing regions, especially in East Africa, freshly harvested sweet potatoes that get even slightly scraped or skinned lose a staggering 8 to 30% of their weight to rot and water loss in just 2 weeks. Think about that for a second. Imagine losing nearly a third of your entire harvest simply because the skin got a little dinged up during handling. It's a tangible, truly devastating loss of life-sustaining food.
Which brings us to Section 1, the sweet potato skin problem, a global agricultural challenge.
Now, this skinning injury is a huge roadblock, literally everywhere. We're talking storage and transport nightmares in developing nations, all the way to struggling with the harsh, bulk, mechanized harvesting we use here in the US. But here's the crazy part. Historically, breeding programs were chasing totally different goals. They wanted higher yields, that perfect, consistent spindle-like shape, and disease resistance. They actually intentionally avoided developing tough-skinned lines because, well, they presumed consumers only wanted a perfectly smooth, unblemished potato at the market. But by chasing aesthetics, they left the crop incredibly vulnerable.
Moving on to Part 2, Hunting for Healing Genes, the biological detective story.
So to solve this major vulnerability, researchers basically had to become biological detectives. They needed to catch the exact healing genes in the act right after an injury happened. And to do this, they set up this really ingenious methodology they called gene fishing. First, they intentionally scraped the thin outer skin off of a specific freshly harvested sweet potato cultivar, LA07146 to be exact. Then, they started a literal 12-hour ticking clock. They took physical samples from the wounded area at 0, 2, 4, 8, and 12 hours. They froze those peels instantly in liquid nitrogen, extracted the RNA, and then amplified the genetic evidence using what's called an ACP system. They were literally tracking the microscopic emergency response minute by minute.
And wow, just look at this. These gel results are the undeniable visual proof of that gene fishing in action. Notice those dark bands highlighted by the black arrows? At hour 0, the band isn't really even there, but by hour 4 or hour 8, it becomes heavily pronounced. What you are looking at is a specific gene leaping into action to save the plant.
Let's get into Section 3, the genetic first responders, securing the scene at the 2 to 4 hour mark.
You see, the sweet potato treats the scrape exactly like a full-blown biological emergency, deploying specialized response units over a highly orchestrated 12-hour clock. Within just 2 hours of the injury, genes named ELIP3 and GDC activate to stop immediate dehydration. By 4 hours, the TAL gene kicks in to begin broader defense responses. Then, at the 8-hour mark, the heavy construction genes, CCOMT and PAL, arrive on the scene. And finally, by 12 hours, the extensin gene thickens up the brand new cell wall. It's a perfectly timed operation. Let's break down exactly what these specific genes are actually doing. Okay, let's look at those crucial first responders, ELIP3 and GDC. When the skin is scraped, the potato's internal moisture is instantly exposed to the air. If the plant doesn't act fast, well, those cells will just dry out and die, a process called desiccation. So the ELIP3 gene jumps in and acts as a protective shield against this devastating damage. Simultaneously, the GDC gene ramps up the plant's stress signals through the GABA shunt. It's effectively putting the entire surrounding cellular area on high alert to secure the injured scene.
What researchers discovered here is honestly a beautiful, highly coordinated relay race happening on a microscopic level. On one side, you have your transient early genes. They spike super quickly at 2 to 4 hours to manage the immediate trauma and send out those alarm signals. But they don't stick around. They gracefully hand off the baton to the late response genes, which show a massive, sustained upregulation at the 8 to 12-hour mark. These late responders, they're the ones responsible for the permanent, physical rebuilding of the plant's armor.
Which brings us perfectly to Part 4, Rebuilding the Cellular Wall, the 8 to 12-hour shift.
Right, so with the emergency stabilized, the bleeding, so to speak, has stopped, and the plant completely shifts its focus to heavy construction. Genes like COMT and PAL practically explode into action right at the 8-hour mark. Just look at the towering purple bars representing the 12-hour time frame here. Gene expression goes completely off the charts compared to where it was when the injury first happened. The plant is throwing absolutely all of its energy into these specific genes. But you might be wondering, what are they actually building?
Well, these highly active genes are essentially the cellular bricklayers. They are rapidly manufacturing two tough, highly protective structural polymers called suberin and lignin. By depositing the suberin and lignin directly at the injury site, the sweet potato creates a permanent, waterproof seal over the wound. Honestly, it's almost exactly like how a human body forms a tough scab over a scraped knee. It's nature's Band-Aid.
And to finish the job, the plant calls upon a gene that codes for extensin. Think of extensin as the final microscopic scaffolding. As the suberin and lignin are being laid down, this extensin gene swoops in and thickens the newly built cell wall, locking everything tightly into place and officially completing the 12-hour healing process. The breach is completely sealed.
All right, our final section. Section 5, Breeding Better Sweet Potatoes, the Future of Global Food Security.
So why does all this 12-hour microscopic drama actually matter to a farmer staring out at a field? Because we can finally apply this genetic evidence to real-world agriculture. Scientists can now use genes like ELEAP3 and CCOMT as clear, reliable expression markers. Instead of planting sweet potatoes, scraping them, and waiting weeks to see if they rot, breeders can quickly screen young plants for these specific, highly active genes. They can purposely breed lines that are incredibly tolerant to skinning. This is a total game changer. It enables rougher mechanical harvesting in the US and drastically, crucially, extends post-harvest shelf life in places like East Africa.
Which leaves us with a truly fascinating question to ponder. By mastering these microscopic genetic markers, could we completely eliminate post-harvest crop loss in our most vulnerable global communities? It turns out, the ultimate key to securing our global food supply might just be hiding in the DNA of a tough-skinned sweet potato. Thanks for checking out this explainer, and keep staying curious.